child or no offspring abnormal. Understand my sentences? I'm saying, are we all born with variations? Yes, we all are born with variations, but we are absolutely genetically normal. क्यों? क्योंकि जो errors था, those errors are hardly 1%. Changes है DNA में, हाँ है. Proteins are different from our and our parents' body, yes, and that is what is responsible for few characteristic changes. But उसमें क्या? Are we becoming genetically abnormal? No, not at all. So when does a child become genetically abnormal? When the errors are much more. Errors in the chromosome, the errors during DNA copying, agar if it is more than 5%, 10%, 12%, 15%, then definitely the infant, the child is born with some genetic disorders. So that is the concept. But what is variation? Is it good for an organism? Is it good for a species? Now try understanding what I am speaking. I am saying variation kya advantages hota hai for these things you get in your questions. Are variations advantageous for a species, for an organism? Yes, majorly. Majorly. Whenever we talk about the small changes that we show in our cells, in our organisms, which expresses in the form of pigments and structures, these variations make us better suited to the environmental conditions. The environmental conditions are changing.
Nucleotides are like attached to the DNA.
What is that?
Nucleotides attached to the DNA strands are based on the outer environment.
No. The change in the DNA may happen because of radiations and etc. etc. So will be much lesser than 1%. What type of nucleotides or units should come and bind one after the other is absolutely a biochemical process which is regulated by the nucleus of that cell.
So like how are they binded together?
They get bonded and come together. That is an absolute genetic message which will be regulated by the nucleus. It's a complete molecular mechanism.
It's like not a series that.
It is a series. Something like, if I now have to talk about this, something like, say suppose I talk about यहाँ पे एक unit है which is something A and यहाँ पे एक unit है which is something T. Now यहाँ पे क्या आना चाहिए था? T आना चाहिए था and यहाँ पे A आना चाहिए था. So by chance यहाँ पे A नहीं आया, rather यहाँ पे G आ गया. So I would understand this is a variation. So there are 4 different type of units. और एक दूसरे के साथ हमेशा they will pair, molecular pairing बोलते हैं उसको. These are mechanisms. So when we are talking about how does the new DNA form, parent DNA में जो भी message है, according to that message the new molecules will come and fit in. The new molecules will come and fit in. So ये है process. But ये जो process है, right now I'm not emphasizing on the process. इसमें process explain करेंगे. It's an absolute different topic. So when we are talking about this, as of now, just understand that there are units which come and get attached one after the other. किस पे based होके the information जो यहाँ पे है, based on the information जो parent strand है. But ये क्या accurate होता है नहीं होता, and then the variation. Now the question is, variation is advantageous? Yes. I'll just give you 2 or 3 instances. You will very much understand from this. Say suppose I talk about a new generation bird. Birds hai, small young birds hai. Now these eggs hatch and the birds come up, and this entire new generation birds, 10 of them, are known to have a longer pair of wings as compared to their parent. But out of those 10, out of those 11, only 10 are known to have the longer pair of wings. One bird is having a shorter pair of wings. 11 में से 10 का long है, एक का ही छोटा है. So what will happen? This is an example. What will happen? How will you conclude with this situation? You have to understand that this 10 birds with long pair of wings, is it a good variation for them? Yes, they may be able to fly long distances, gather food. What about the bird with the short? The bird with the short will not be able to fly long distances, greater heights. So definitely that is a disadvantageous variation. क्या ये ह्यूमन में भी होता है? Yes, in various different type of animals. And this is one example only I am talking about. There are many such examples. I talk about humans. Say suppose I talk about in case of human also, are children born with some small defects in their liver, in their heart, in their eye, etc. So these children are always bit different or bit weaker than the rest.
These defects in the body parts are caused by genetic variations.
Exactly because of this. The reason is this one. So now this child, out of, in one particular generation, I come across 50 children are there. Out of those 50 children, only 5 children has got some defects in the heart and some defects in the kidneys. And now what is happening? These 5 children generally, will they survive quite a number of times till the reproductive age? Quite a number of times they don't survive. Because defects in the brain, defects in the heart, defects in the kidneys, they may not survive till the reproductive age. तो इसीलिए ये वाले जो variations है, why are they not common in a population? Because most of the time people born with defective variations, they don't survive till the reproductive age. And if I talk in terms of human, you will understand much better. These are those few individuals who would not lead a normal life. So there is no chance. How will this variation pass on to the next generation? What about the rest 10? They are leading a normal, healthy life. They get married. They pass on the characteristics to the next generation. What about this 5? No, they are not leading that normal life. Maybe they are not surviving till the reproductive age. So there's no chance why they pass on the characteristics to the next. What are we trying to understand?
If a family has a diabetes disorder, so it will pass on to their children.
Definitely. Diabetes is quite a simpler condition in respect to this. When we are talking about children born with defects in the eye, defects in the heart, vital organs, heart, brain, kidney, vital. Most of the time, they are dying out at an age of 15, 16, 18, 20, 23, much earlier as compared to the other organisms. So definitely these characteristics are not passed on. You get questions, are variations beneficial for the species? Are variations beneficial for an individual? More than an individual, variations are beneficial for the species, the whole lot. How do you understand a variation? You will get certain case studies, some examples there. How do you understand variations are, how do you understand from two variations, two changes in organisms, how do you understand that a specific variation is advantageous? Advantageous variations will always be spread out in the population. Yes or no? And जो भी disadvantageous variation रहेगा, whatever I had discussed so far, what did I tell you? That if it is a defect in the vital organs or if it is a shorter pair of wing and etc., these characteristics क्या ये pass on हो रहा है to the next generation in large numbers? नहीं हो रहा. But this long pair of wings will get passed on to the next generation very smoothly, very easily. So how do we understand a variation advantageous है? If it is present in large number of organisms of the species. How do we understand? एक variation disadvantageous है when it is present only in a limited number of individuals in a population. ये बात आया समझ में? So these certain questions we will do later. So this is first what are you to understand? How variations, why variations, and are variations advantageous, disadvantageous? Now next is we try understanding the process of reproduction in different group of organisms. So next part that we are going to take up is reproduction in, we have the volume. This is volume 2. Volume 1. Now, next is we are going to talk about the concept of variation. See there in page 20, the first page, all of you turn to. Look into page 205. There is a question. This is very important. As it is, you may get this in your paper, also school paper. Why is variation beneficial to species but not necessary? 205. See there, there's a question. Why is variation beneficial to species but not for the individual? This is what I told you just now. Next is we move on to the modes now. What are the two types of variation? The two modes of variation. One would be, we now talk about the modes of variation. What are the two modes of variation? Number one would be
Asexual.
Asexual, and the next would be sexual. So we are talking about the two modes of variation. What are the two modes? Number one is asexual. We talk about asexual reproduction, and the second is sexual reproduction. Make a table. You will have to write down some major differences between sexual and asexual, and then we will start with the asexual modes of reproduction. What are the organisms and what are the methods?
But what is the need of continuous variations?
What is the need of?
Continuous variation.
There is no need. If inside our cell the biochemical methods would have been accurate, then there would not have been any variation. तो पहली बात तो ये है, हमारे बॉडी के अंदर जो सेल के अंदर जो बायोकेमिकल प्रोसेस है, that we are not able to regulate. It is a spontaneous process. वो 100% accurate कभी नहीं. So this is one reason. Second, क्या variation क्यों होता है? So definitely variation इसीलिए हो रहा है because every generation is better adapted to the changing environment. You talk about yourself. The way you are adapted to this environment full of pollutants, your grandparents and another 5 generations back, they will not be adapted to this changing environment. They find it very difficult, and they are finding it very difficult. But for us, are we finding it that difficult? No. You all are much more comfortable. The next generation more comfortable because we are born in this type of a situation. So our different type of enzymes and hormones and pigments make us get accustomed to this situation. Even in the fishes in the water, the birds in the aerial environment, every type of a sexually reproducing organism is born with small changes. And the changes are as per the changes in the environment suit better. When you are asked questions, why variations are a necessity? Variations always make an organism better adapted to the changing environment. Which is not fit, or this changing environment is not fit for the 3 generations prior organism to survive. 5 generations back, organisms find it very difficult to survive in this changing organism. We can easily understand this when we compare ourselves with our grandparents, or if some of you have your great grandparents still alive. So you must be very much realizing that how the differences that they talk about and what are some of the difficulties that they face.
So like these variations can also be caused by errors.
Allergy.
No, variations can also be caused by errors.
Variations are caused by errors.
Like error is actually caused by errors, but then the variation happens due to like there are already errors in
the process.
So there might be an generation.
जहाँ पे error हुआ ही नहीं. अब तक तो नहीं. So far we always have come across, is any generation born with DNA? कैसे हमें पता चलेगा that in this particular generation or in this particular parent, offspring or parent के बीच, when there is absolute similarity, then perhaps we will be able to understand, हाँ, इस parent में or the DNA जो went in the formation of the offspring, the son and the daughter, कुछ changes हुआ ही नहीं होगा. So for that, that son or the daughter should have all the characteristics, half or some match with the mother, some match with the father. So far, in the concept of genetics also, whenever we study, the theories have not changed. आगे चल के God knows, maybe हो भी सकता है, but error होने का ही chances ज़्यादा है, correction होने का chances तो वैसे भी कम ही है. So when we are talking about errors in the process of duplication of the DNA, will keep on increasing even more. Because the process of formation of the new species is very close. When we are talking about ourselves also, so much of changes have already added on. तो correction कम होगा या errors कम होगा, उसका तो chances बहुत ही कम है. Errors बढ़ेगा और बढ़ेगा because so many different type of factors which are changing the DNA. So what are some of the differences you should know? Sexual and asexual. Make a table very fast. And let us write down some major differences, sexual and asexual. For asexual, number 1, you write. Only one parent is involved in the formation of. Only one parent is involved in the formation of new generation offsprings. Only one parent is involved in the formation of new generation offsprings. Other side. Two parents involved in the formation of new generation offsprings. Two parents involved in the formation of new generation offsprings. Number two, asexual. Number two, for asexual reproduction, large number of offsprings are formed. Asexual. Large number of offsprings are formed. Numerously at times. Large number of offsprings are formed. Large number of offsprings are formed. Sexual, few offsprings are formed. Few offsprings are formed. Few offsprings are formed.
The DNA tests are not 100% correct.
The DNA tests
Which are conducted are
not accurate. Not absolutely not accurate. But in the DNA test also, what are you understanding? And from the DNA test only, it is very much valid. The DNA test show child and the father, the child and the mother, when the DNA is matched, it is never found that it is 100% matching. Which maybe it is found, and from that only the concept. No, but it is understood always that there is 5 to 7% of a change always in the offspring's DNA from the parents' DNA. Now, when we are talking about, continue the next point, what is the next? Few offsprings are formed for sexual reproduction. Number three, for asexual reproduction, no special reproductive cells or gametes. Special reproductive cells or gametes are not formed. Special reproductive cells or gametes are not formed. Other side, sexual reproduction. Special reproductive cells or gametes. Special reproductive cells or gametes are formed. Special reproductive cells or gametes are formed. Last point, asexual, number 4. Number 4. Offsprings are mostly identical. Asexual. Offsprings are mostly identical. Offsprings are mostly identical. Offsprings are mostly identical. For sexual, offsprings are with variations. Offsprings are with variations. Offsprings are with variations. Now, go to the next page and start drawing these diagrams. Very simple. With pencil, you will draw. All of you try doing it very fast and just learn 2, 3 features. This diagrams, entire set of diagrams, are very, very important. 2-mark diagrams definitely there in the paper. So you should know this. Get started with the modes of asexual reproduction, one after the other. We are talking about, as and when I discuss about the points, the feature, 1 or 2 points you should know. The advantage, disadvantage, the differences you should know. And what are some of the characteristics of the method you should know. I'm talking about the process of asexual reproduction. Now, in the case of asexual reproduction, the first method that we're going to talk about would be fission. The first method that we talk about is fission, and there are 2 types of fission we are going to study. One is binary fission. One is binary fission, and the other one is multiple fission. So now, We start with the first method. What is this first method? This is binary fission. This is binary fission. So now in case of binary fission, what happens? Look at the diagram. What happens in binary fission? Very simple. Single cell organisms will always show this method. Examples you'll always have to remember. Amoeba is there. Then we talk about euglena is there. These are different type of organisms. Simple cell organisms, amoeba, paramecium, euglena. Very much show this type of method. What is this method about? It's a parent single cell, and this entire set of diagrams you get for rearranging also in a specific sequence. What is the method? The parent organism is there. Next, the cell elongates little bit, followed by the elongation of the nucleus. What is the third step? The constriction in the nucleus, followed by the division of the cytoplasm and the two new daughter organisms. Very simple a method. Very simple a method which takes place in single-celled organisms. How many organisms formed at the end? Two organisms. Two daughter organisms formed at the end. Are they exactly identical to the parent? Yes, they are exactly identical to the parent. Is the parent still existing when the two daughter cells are formed? No. The parent is not existing when the two daughter cells are formed. So what is that point you should always write as a difference with other method? The parent identity is lost. The parent identity is lost, meaning what? The parent identity lost, meaning it is the parent body only which got divided into two new offspring. Let us talk about the second method. The second method of fission, that is known as the multiple fission. We are talking about multiple fission and what happens in multiple fission. Let us try understanding. In the case of multiple fission, here the difference is it is again a single cell organism we are talking about, but this single cell organism, majorly these are pathogenic forms, meaning the disease causing ones. Amoeba is not disease causing, no. These are the free living organisms, amoeba, paramecium, euglena, all these organisms that you talk about, these are all free living. They don't cause any harm, they don't cause any disease. So what is the second type that I have to talk about? And this is known as multiple fission. In the concept of multiple fission, what are we supposed to understand? Examples, say suppose I talk about organism, and this is the plasmodium. The plasmodium, the trypanosoma, this type of organisms, these are all the disease-causing organisms. And plasmodium, in reference, what type of a disease they cause? It's a protozoa, plasmodium. You must have come across. Malaria. So when we are talking about this particular protozoa, single cell organism is responsible for causing malaria. But how does it divide? It divides very fast in a huge, large number. What happens? It will now divide into a large number of nuclei like this. The nuclear division is multiple sites. See here, initially we were talking about binary fission. The nuclear division is just once. But now here, multiple nuclear division. What is now happening here? This is multiple nuclear division takes place, and as a result of multiple nuclear division, what are we going to call this as? These are known as nuclei. Next, what would be the next stage? The number of nuclei will keep on increasing as long as the outer membrane does not burst up. So here what will happen is the entire number of nuclei, with huge number of nuclei will start getting formed, and after a particular period of time, every single nuclear fragment will be covered up by some small amount of cytoplasm, will be covered up by a small amount of cytoplasm, and that is the small new young organism that is getting formed. So every single small amount of the cytoplasm is getting wrapped up by a small amount of cytoplasm like this, and this results in the formation of the young plasmodium, the young offspring, the young new generation organisms. So this new young generation organisms, they will all get released. How they will get released? By the rupture of the membrane. So what happens here? After a particular period of time, all these nuclei, they will all rupture, the membrane ruptures, and they would all get released out. So what are these? These are the young new generation organisms, or these are the new generation or organisms or the offsprings, also known as the, you can also write down the daughter plasmodium, that also can be written, or the new generation organisms also can be written. What is happening here? Multiple nuclear division and rupturing means breaking down, rupturing of the wall of the parent cell, the membrane of the parent cell, and the release. This method of multiple fission is very rapid, and as a result, the process of infection. Just now I told you that this is an organism which is responsible for causing a disease in our body. So that's the reason, since the number of nuclear division is so many, a large number of such small cell-like organisms are formed, and they are released in large numbers, and then they mature after a particular period of time. That's the reason the infection spreads so fast. Now tell me, what are some differences you can mention between these two methods, binary and multiple fission? Here the nuclear division is just once. Nuclear division, numerous, multiple times. What happens here? In this case, multiple daughter cells, only two daughter cells are formed. This is responsible for causing diseases, various different type of infection, multiple division. Organisms growing by multiple division are generally responsible for causing some of the other type of diseases, and these type of organisms dividing by binary fission are generally not pathogenic. They are not responsible for causing any diseases generally. Yes.
At the school today, the teacher told that plasmodium is amoeba in unfavorable condition.
Plasmodium is amoeba? I remember. Plasmodium is not amoeba. Plasmodium and amoeba are absolutely two different organisms.
No, she said that under unfavorable conditions, amoeba forms a cyst around itself.
No. The amoeba doesn't form a cyst. This forms a cyst. This
she said that under unfavorable conditions, amoeba binds into a circle, forms a cyst, and that thing is called plasmodium. She just told us today.
इस पे मुझे भरोसा नहीं है, सुनता नहीं अच्छी तरह से. Plasmodium is absolutely different, which causes malaria. It's a protozoa. This is what is causing malaria. The method of division is multiple fission. Where the nucleus, हाँ, अगर वो चीज मुझे बोलना है, वो भी मैं बोल सकती हूँ, but that is not much necessary. अब इसमें क्या हो रहा है? Now that they are released, say suppose something like this, what is happening? They are getting protected by some additional wall. They are getting protected by some additional wall. This additional wall को क्या बोलेंगे? Cyst wall. So what is the cyst wall? This is now, if I talk about this, then this you can write as a difference also. In binary fission, the two daughter amoebae, they are not protected by any cyst wall under unfavorable condition. But now say suppose this membrane ruptures, the temperature is very high, humidity very high, not suitable for the growth of the plasmodium organisms. What will happen? They will remain dormant for a long period. जब तक conditions favorable ना हो, they may remain dormant, and जब conditions favorable हो, they will grow again. Amoeba is an absolutely different group of organism, free living है. Plasmodium is an absolutely different group of organism, non-living, non-pathogenic, pathogenic है. This does not form cyst wall. इसीलिए when the amoeba reproduces, unfavorable condition अगर है, both of them will die away. Plasmodium, why are they so resistant? क्योंकि unfavorable condition है तो they are not dying, they are just becoming dormant for a short period of time. So these are the differences that you will write. What is the next method that we talk about? Asexual reproduction. The next method that we talk about would be fragmentation. Let us talk about this. What is fragmentation? Now, in case of fragmentation, we are majorly talking about organisms like Spirogyra. We are talking about organisms like Spirogyra, organisms like Eulothrix, etc., etc. Where are these organisms present? These type of organisms are present in the pond, lake, shallow water bodies. And how would their general structure be? Their general structure would be, they would be generally filamentous like this. They would be generally filamentous. And of course, these are multicellular structures that we understand. These are multicellular structures. And these are plant-like organisms. Why do we say plant-like? Because they have some chloroplast in them, cell wall, etc., etc. So these are filamentous algae. Basically, they are known as algae, Spirogyra. Spirogyra, Eulothrix, these are filamentous algal forms in the water bodies. And this Spirogyra, when mature, what is the name of this organism I am talking about? This is the Spirogyra I am talking about, which is a multicellular organism. Under natural conditions, not accident, remember, try understanding what I'm speaking. This is the entire organism. What is this filamentous structure known as? This filamentous structure is known as the thallus. Thallus, ये जो पूरा filamentous body होता है, इसको thallus बोला जाता है. Now, when the organism is fully mature, इनमें कितने segments होते हैं? At the most 20, 25, 50, उससे ज़्यादा नहीं होता. They are not complex organisms. हाँ.
Are these organisms capable of carrying out photosynthesis?
हाँ, chloroplast है.
It's an algae.
Algae. It's an algae. Algae can carry out manufacturing food. Algae can carry out photosynthesis. It's a plant. Actually, these are thallophyta. Belong to the group thallophyta. So when we are talking about this Spirogyra, how do they divide? They would, now that the parent body had fully matured, what will happen? The entire parent body will now divide into certain fragments naturally. The entire parent body divides into certain fragments naturally. The entire parent body will divide into certain fragments naturally, not programmed, just at random. Now, if the parent body has adequate number of dividing cells, then this will grow and complete the entire structure again. This will grow and complete the entire structure again. So here, generally we come across that how many new Spirogyra should be formed from here? 3 new Spirogyra should be formed from here. So what are the features I'm talking about? The features that I'm talking about here is fragmentation is a process where filamentous algae naturally divide their body into certain parts. Naturally. So ये उनका process of reproduction ही है. It is their method of reproduction. So जब पूरा filament बन गया, thallus जब बन गया, automatically the filament will break up into few fragments. अब ऐसा भी हो सकता है कि there is one fragment इतना सा. There is one fragment only this much where adequate number of dividing cell नहीं है. Will this give rise to a complete new organism? No, this will not. So sometimes you get figures. एक Spirogyra है, उसके 4 fragments है, एक fragment बहुत ही छोटा सा. And questions are asked: how many new Spirogyra will be formed from this? How many new Spirogyra? Now you have to check the filament. वो filament में fragment में अगर adequate number of cells है, and they will divide it completely, form the filament. The filament is very small. The fragment is very small. What are these? These are fragments. Why the method known as fragmentation? Because the mature organism divides the body into few fragments naturally. इसीलिए method को fragmentation बोला जाता है.
Multiple fusion में parent identity है.
Lost. Parent identities, these are the similarities when you are asked to write. You are asked in the exam also write one similarity, one difference between multiple and binary fission. So even in fragmentation, parent identity is lost. Okay. So what is happening in fragmentation? Here we are talking about a process where the organism, the body of the organism is dividing into certain fragments, but naturally, not under any accident. And this results in the formation of certain organisms. Next is we talk about another method after fragmentation. We talk about another method known as spore formation. Now, spore formation is a method where we come across this type of a method generally helps in the variety of fungi. Spore formation is a method of reproduction taking place in the fungi, the kingdom fungi. What are some of the examples? Kingdom fungi, you can remember the mushroom. Mushroom, Eurothrix है, Anubina है, all these are organisms. Now, book में I think Spirogyra एक ही example दिया हुआ है, देखना what are some of the other examples there. Now, next is we talk about these methods, spore formation. Where are these organisms taking? Where are this method taking place in fungi? Now, fungi. What some examples you are supposed to remember? You can write down examples of the mucor. You can write examples of rhizopas. In your book, example of rhizopas is given. Yeast also, no, yeast is budding. Yeast fungi hai, but that is a unicellular fungi. I'll tell you that. So, पहले we will do this. The rhizopas, that is the bread mold. Bread के ऊपर जो mold होता है, देखा कभी?
Yes.
So that is called the bread mold. क्या हो गया? It's kind of अभी हो रहा है. Then standard, थोड़ा बैठने की आदत डालो. Now, when we are at home, how long can you sit and study at a stretch? 2 घंटा. हाँ?
2 or 3 hours.
2 to 3 hours. 1 hour. Kindergarten में हो अभी 1 hour. Now, when we are talking about rhizopas, what is the method? Say suppose I talk about the structure of the rhizopas. How will the structures be? Look into this. The process of spore formation. How does it happen? First of all, we are talking about the structure of this fungus. Which example am I talking about? The bread mold of the rhizopas, which is growing on the surface of the bread, on the surface of the citrus fruits, etc. And they have got this filamentous structure. What is the name of this filamentous structure? This is known as the hypha. So these are all multicellular structures. Remember, these are all multicellular structures. Nobody will wind up. Huh? We will complete with this and then wind up. Now, these are multicellular structures. What are these multicellular structures known as hypha? And what is this network of the structure at the base of the surface known as, or the substratum known as? This is known as mycelium. This you will have to remember. Hypha, how will you define hypha? How will you identify? Hypha is the filamentous structure, multicellular filamentous thread-like structure. And what is mycelium? The network of those thread-like structure on the surface of the bread, fruit, decomposing food anyway. Now next what happens? This particular hypha, some of them will start showing the process of cell division, and it is not all the cells, but one particular group of cells only will show. One particular group of cell will show the process of cell division. So now what is happening exactly? Here, this multicellular structure, the hypha that I told you, only one single cell will start showing multiple nuclear division. And now in that particular cell, something like this will only show the process of multiple nuclear division. So now when this shows the multiple nuclear division, a knob-like structure gets formed. Numerous nuclei there. But what are you going to name this nuclei as now? These are known as spores. What are these multiple granular structure getting formed inside known as spores? So what is the name of this structure now? The name of this structure is sporangium. So what is that knob-like structure in the rhizopas or the bread mold known as, which contain maximum number of nuclei or the spores? Sporangium. So once the number of these spores become too many, what will happen? The sporangium will burst out. The sporangium will burst out and all these spores will be dispersed off. The sporangium will burst out, the spores will disperse off. Does fungal infection happen very fast or slow? Very fast. Why? because these spores can travel long distances, short period of time, because they are very small and extremely light they are. So they can travel long distances, and on top of that, just like the plasmodium, they also can develop a protective sheath around something known as the cyst. So under unfavorable conditions, immediately the spores do not get destroyed. They can survive, withstand unfavorable conditions. So that's the reason getting a cure or clearing up any fungal infection is very difficult. How many methods of asexual reproduction we had studied, and these are the main methods of asexual reproduction. What did we study? Two methods of fission, fragmentation, and spore formation. The next class we are going to study what? Regeneration, which and budding. Okay, budding is also left, and that is different. Regeneration, budding, another true method of reproduction left. And next we are going to study regeneration, which is not a method of reproduction, but only a method of regaining the lost part. We'll continue with this the next class. Which one is your book? Is this your?
दुनिया में 40 जन तो मूव कैसे
I was